what VEXAS syndrome is and how this might be related to myelodysplastic syndromes. We hope today's session brings a little bit of clarity and answers to your questions. So with that, I'm really happy to introduce today's speaker, Dr. Tara Lasha. She is an assistant professor of medicine with the Division of Hematology in the Department of Internal Medicine at Mayo Clinic in Minnesota. And I'm really, really happy to have her here. So the room is yours. Thank you, Dr. Lasha.
Thank you. Thank you, Mary Lou and HealthTree for the opportunity to present today. I'm going to share my screen. Okay, can you see it okay?
Yeah, perfectly.
All right, laser works. All right, so thank you everybody for the opportunity to present. So today I'm going to tell you a story about a new disease called VEXAS, and this disease was just discovered in 2020. And even though this story is specific for one disease, the findings that we have acquired since 2020 have implications for how we study and understand other heme-related malignancies. And I want to make clear that I'm not a medical doctor, I'm a research scientist. So I won't be focusing on symptoms and treatment, but more on the interesting science, as we learn about this. So I'll start the presentation with an introduction and a background into acquired mutations and clonal hematopoiesis. Then I'll talk about VEXAS and how it was first discovered in the blood and some of its symptoms and features. Then I want to talk about the gene UBA1 that is mutated in this disease, why it's so interesting and what we're learning about it and the pathway that it controls. And finally, I'm going to discuss a bit about the heterogeneity that we'd see in the disease, and I'll share some of the research that we're doing, which is actually really helping us understand the complex associations between things like myelodysplastic syndrome and adult onset inflammatory conditions. So my passion for this comes from being a part of a team that runs a pre-myeloid bone marrow failure precision medicine clinic at Mayo Clinic. So this clinic began in 2017, and we started this as a home for patients that have abnormal blood counts or bone marrow failure symptoms who don't necessarily have a diagnosis. So they're not, put in one specific thing. And these patients tend to be shuffled around to different departments. And so we really wanted to be able to take their precious blood and bone marrow that they were willing to donate and provide all the tools and services and direction that we could for these patients, the patient that walked in, right at that moment and during their treatment, rather than bank it and do retrospective studies and whatnot. So I feel sometimes we're so siloed into specific cancer syndromes, that patients feel really helpless of, what can they do? They don't have a diagnosis, but they clearly have something wrong. And so some of the criteria of the patients that we get, they might have a mutation in the blood compartment in kind of a lab result that they got back that they don't really understand. Or a screening, they could come in with an unexplained low blood count, and they don't know why. Or maybe they had germline testing for cancer or a hereditary syndrome, and they have a result that isn't really conclusive for a germline, but it could be an acquired mutation, and it could have something to do with hematopoietic defect. We test patients prior to specific chemotherapeutic treatments because sometimes we find alterations that we know will immediately lead to a therapy-related myeloproliferative neoplasm if they're exposed to a specific therapy. And then finally, we screen, prior to an autologous hematopoietic stem cell transplant. So interestingly, most of our referrals come from outside of hematology. So these patients present in rheumatology or the breast clinic or, somewhere else. And, what's important and underscores, the complexity of multisystemic involvement in these diseases is that these patients have a plethora of symptoms. And every patient is unique. So they have unique phenotypic characteristics. And so this is a great example of how precision or individualized medicine can be used for care. So we take these patients, we run a very extensive gene panel analysis on them, and then what we find at that moment, we curate the variants using a bunch of different techniques, and then we assemble as a tumor board. So, and we provide education and expertise to the different silos in cancer and different practices, as to how to treat and advise the patients as they come through. So that being said, as we age, we're all inherently susceptible to direct and indirect DNA damage, repair, and replication genotoxic exposures. And when this happens in the blood, this is called clonal hematopoiesis. And the potential of this, most of these are just passengers, but the potential is this is a fitness advantage for the cell, which can allow for clonal expansion. These cells can increase their proliferative drive. They can self-renew if they're stem cell at the stem cell level. They have an improved ability to evade death, and over time, these expanded clones can influence human health. So clonal hematopoiesis is a normal age-related phenomenon, although it involves genes that we typically see in leukemia. However, the vast majority never develop a leukemia. But what's really interesting, and I guess intuitively expected, is that the presence of clonal hematopoiesis is increased with all-cause mortality. It's seen in various different diseases, coronary artery disease, stroke, and there's also associations with things like diabetes and osteoporosis, and there's still a lot we have to learn about the presence of this and the context related consequences in patients. So now let's talk about VEXAS. So this slide is pretty busy, but we already know a lot about inflammation because over the past 3 decades, as I'm showing here, and with expanded and increased utilization of genetic testing, we've really had the fortune of characterizing a lot of inherited autoinflammatory syndromes, through assessing familial clusters. So we're seeing phenotypes. So we see a family, all the family is suffering from the same kind of inflammatory features. And so we can test them and we can compare the testing between the family members and come up with, the gene that's responsible. So I have 39 different diseases listed here with their causative gene, inflammatory feature, and the mechanism of the disease. So these are pretty relatively easy to spot. However, what about cases that have abnormal inflammation that don't have a detectable germline variant, or they don't have a family history? Is it allergies? Is it too many jalapenos in the salsa? Or is it really something serious? And these can be really hard to characterize. And now, since I just told you there's an incredible complexity of acquired mutant states in the body, and sometimes these states have phenotypic associations or symptoms that kind of correlate with them, this is particularly relevant, and especially for patients that have, later onset disease manifestations and that don't have, affected family members. So this thought process is what led to the discovery of a syndrome in 2020 called VEXAS. So the group at the NIH had access to a large genomic data set, and these were patients that had undiagnosed disease. It was a large cohort, 2,500 cases. So these were protein coding DNA regions that were mapped. So this group screened for novel alterations. They focused on the ubiquitin pathway, and I'll talk about that in a little bit, and it's very interesting because a lot of autoinflammatory diseases that have been discovered with genetic alterations are in this pathway. There's almost 840 genes in the pathway. They were looking for a pathogenic variant, a novel one, and not reported in any human healthy databases. And as I have shown here from the screen, they identified 3 male cases that had missense mutations in a gene called UBA1. All of these men presented later in life with onset inflammation. And shown here are the PCR sequences of the mutations. So each peak represents a nucleotide, and you can see here by the arrows shown that there's a base change. So it's a C to a T, a G to an A, or a C to a T here. And so the men resulting in a double peak. However, this gene UBA1 is actually on the X chromosome. And these are all men, so they should have only one copy, so that we shouldn't see a double peak here. And so this is telling us that some of the cells in the blood have this mutation and some of them don't. So this is an acquired mutation. So the group isolated and sequenced different cell populations in the blood, from 6 different cases that they then identified. And then they showed that this was indeed only found in the blood and the bone marrow. The patients also had wild type lymphocytes, so T cells and B cells did not have the mutation. There was no presence of the mutation. Instead, this was restricted to the myeloid cells. So these are the innate immune cells. These are the cells that get really angry. They release the histamines. They react right away. And these mutation burdens are really high. And the group also sequenced skin fibroblasts, which were negative. They're right here. And this validates that this is not a germline mutation. This is indeed an acquired mutation. So this group named this disease VEXAS syndrome, and these are the salient clinical features. It's an acronym. So V stands for vacuoles. So these are the little white dots that you see here, and they're in the marrow. E is for the E1 ubiquitin activating enzyme, which is encoded for by the UBA1 gene. X stands for UBA1 X chromosome, as I mentioned. A, these patients all have autoinflammation, and S, the mutations are somatic. They're not germline, they're acquired, they're somatic. So how do you recognize this disease? Well, the most obvious thing is that it's predominantly a disease of older men. And this is because there's only one X chromosome. Women have another X chromosome, and so this can compensate for the mutation. However, we do have a case of monosomy X that has VEXAS in a female. Since this disease was discovered only in 2020, we're still, needing more screens on female cases in order to get a true, representation. Patients commonly present with high fevers and a dependence on steroid treatment. The disease results in severe inflammation, but I have to underscore this is a very highly heterogeneous disease. So it can present with both rheumatologic and hematologic conditions. It's quite interesting because if a patient is seen in rheumatology, they might get diagnosed with a rheumatologic disease, and if they're seen in hematology, they may get diagnosed with a myeloid neoplasm. So it overlaps. These patients have a predisposition to hematologic conditions. So 30 to 40% have MDS, some have MPN, myeloproliferative neoplasms, lymphoproliferative neoplasms, plasma cell dyscrasias, including multiple myeloma or MUGUS, and a lot of these patients have recurrent thrombosis. So this is a picture of the VEXAS male. They can get fever, they can get inflammation in various structures in the head and neck, the lungs, the gut, all this bone marrow pathology. They get inflammatory arthritis, they get a whole range of skin disease, and a very high rate of thromboembolic disease. So over 40% of patients have either arterial or venous clotting. So this is really heterogeneous disease. So common features in the disease, patients have high fevers, they require steroids, there can be lung involvement. So this is, lung, this is the pleural fluid around the lung is full. They have vasculitis in the arteries, neutrophils in the alveoli, so this is in the lung. The striking characteristic finding are these vacuoles in the erythroid and myeloid precursor cells. And so these are these little white dots in the cytoplasm of the cells. This has become a really sensitive finding for this disease, but not every patient will have it. And it's not a fully specific finding because you can have like a copper deficiency. This is associated with the same type of phenomenon. These patients typically have skin involvement, so angry skin, vasculitis on the, cartilage of the ear, the nose, the eye. And on the right here, there's the presence of this macrocytic anemia here, so these large red cells that are kind of pallid in color. This is a very frequent 90 to 100% of cases. They have an increase in neutrophils, neutrophilia, but these are left shifted, so these are immature forms. So this is telling us that the blood is requiring more to be produced, and this is because there's an upset in these cell types here in the blood. This is a myelocyte, and down here below there's bone marrow, and the two circles show some left shifted or young erythroid and granulocyte immature cells with vacuoles. So what does UBA1 do? This is the gene that's mutated in this case, and this is super interesting. This is the ubiquitin proteasome system, so the UPS system. And this pathway actually involves delivery of a cargo. So this yellow kind of ball here is called ubiquitin. Through a cascade of enzyme regulation, it places it on a protein that's no longer needed, and it kind of tags that protein to go to the proteasome. And the proteasome is kind of like a recycling center that chews it up, spits out the peptides so that they can be reused again. And I know you're saying, oh my gosh, Tara, why are you focusing on this? It's just a recycling system. But I'm sure everybody has had one incident in their life where they've had the trash man not come, the garbage system not work, or recycling, and after that happens, havoc can wreak loose. But to be, joking aside, this system right here actually influences all of cellular homeostasis. It's very important. Cell signaling and protein localization, it's just crucial. And there's 3 steps to this. An E1, an E2, and an E3 step, and this is according to the hierarchy levels of enzyme regulation. So the E3 step here, I've already talked about. There's over 600 of these enzymes, and this is where a lot of the genetic diseases have been discovered, and it's why the NIH group focused on this pathway. These E3 targets thousands of specific substrates for degradation or changing signaling pathways. If you go up a level, there's only 40 enzymes that control this. But at the level of E1, there's only 2 enzymes, UBA1 and UBA6. These are 2 genes, and by far and away, UBA1 does 99% of the work. So this is an acquired mutation in the master enzyme of the UPS pathway. And that's why this is only a somatic disease, because if this was germline, if you were born with this mutation, it would be lethal. And, the most important thing on this whole slide, I want to point out the genes in this pathway are not typically included on MDS gene discovery or diagnostic panels typically. So this is important, and it underscores our need to be able to, look at these different pathways now that aren't historically in typical cases. Even though inflammation is a common feature in MDS, and we know now that 30 to 40% of cases involve MDS in VEXAS. So this is very important. So now let's talk about these mutational consequences. So how are these mutations actually affecting these protein? And this is, I think, important too. This mutation is only in this protein, but it highlights how complex these mutations can be in various spots in the mutation. So panel A here shows the full-length protein of UBA1 with some of the known mutations. So UBA1 has 2 proteins that are produced, 2 isoforms. These are shown in panel B. So there's UBA1A, which is located in the nucleus, expressed in the nucleus, and UBA1B that's expressed in the cytoplasm. Now, if there's a mutation at the position M41, which is the most prevalent mutation, UBA1B is not produced anymore. And instead, there's a cryptic isoform that is now produced. But this one is catalytically impaired, so it's defective, and it replaces UBA1B in the cytoplasm. And this leads to the loss of cytoplasmic ubiquitination. So that little yellow ball can't tag the proteins for degradation any longer. And you can see on the right here, these are protein plots. So it's levels of protein in these patients. The patients are in red here. And you can see that now they have this new expression of this UBA1C. And this results in less of the ubiquitination. So this is polyubiquitination. So this is, typically there's more than one of those yellow balls that are placed on the protein. And now they don't have that opportunity. There's less here. And there's an increase in stress markers. So this drives inflammation in the cells. And in panel C here, this is also important. So in other myeloid and lymphoid malignancies, now that this disease has been discovered, there's been other mutations in UBA1 that have been described. And you can see these mutations involve, intron exon splice areas. So this is actually not in the coding region, but it affects how this is spliced. So this actually takes away UBA1B again and only produces UBA1C, even though it's not in the same position as this most prevalent mutation. And then here on the right, mutations that don't actually affect the isoform at all, but instead influence other aspects of this protein. So one of these mutations actually interferes with ATP binding, so the kind of the energy that's used in order to help this pathway, this enzyme function, or there's another mutation that actually affects the ubiquitin transferability, so the ability to put that yellow ball onto a protein, or maybe there's some more here that I won't go into because it's the median age of onset in VEXAS is around 64, so right in here. The youngest patients described are around 45, and by community data prevalence estimates, this disease is found in 1 in about 14,000 individuals. However, in men older than 50, it's found in about 1 in 4,000, so it's very prevalent. And the fact that we've only found this in 2020, I think really highlights the fact that, we don't have the right kind of surveillance in order to capture these patients. And so this disease itself is really, really providing clarity into, how we need to look at these and monitor and screen for these types of diseases. The penetrance is around 100%, so that means that all individuals that have a UBA1 mutation have a disease. And although the phenotypes are very diverse, the disease is associated with substantial morbidity and mortality. Treatments for VEXAS include here on the right, corticosteroids are typical, and cytokine inhibitors, hypomethylating agents have been used, and also allogeneic stem cell transplants. And although hematologic conditions are common, and the conditions are known to progress, there's no increased risk of AML associated with VEXAS. So we know that UBA1 mutations in the blood lead to VEXAS, and we know there's a lot of different clinical manifestations of the disease. So what determines the heterogeneity of this disease? Why do some patients have severe disease, and why do some get MDS? Why doesn't this lead to acute leukemia? What drives this inflammation, and what governs the mechanism of this disease? So now I'm going to go into current research, and this is really exciting what we're learning, and it's translating to all aspects of heme malignancies. So we, in the collaboration with the NIH, looked at the prevalence of clonal hematopoiesis in these cases. So perhaps the reason that patients went on to having MDS, the 30 to 40%, rather than just rheumatic symptoms, was potentially the association of these co-occurring mutations. So we looked at the rate of percentage of clonal hematopoiesis, and as you can see here on the left, there's more clonal hematopoiesis mutations in VEXAS than in control. So this in blue than in red, which makes sense because this is an inflammatory disease. Everything is kind of ramped up. But when we looked into which genes were mutated, we found that the most common mutations were typical age-related CH mutations. So in DNMT3A and TET2. But what was really surprising is that when we stratified our patients, so each column here represents a patient, by whether or not they had an MDS MPN or whether they didn't. And when we look down here, these are the CH mutations that are associated with these cases, and we didn't really see an enrichment of CH in the MDS or MPN cases. So this suggests that it's not that there is a secondary mutation that might lead to MDS or an MPN. We next looked at what factors were significantly associated with mortality. And in this study, clonal hematopoiesis mutations like DNMT3A or TET2 alone, or DNMT3A hotspot mutations, have the highest risk in terms of the hazard ratio for mortality. So if you had these mutations, you had a higher likelihood, of an inferior outcome. So even though clonal hematopoiesis mutations don't confer more of a risk for MDS, they do increase overall mortality risk in VEXAS. And things like the level of UBA1 mutation in the cases, like every single cell has it versus only 20% of the cells, that wasn't significant at all. So how is the presence of these normal age-related markers contributing to an increased risk for mortality? So a lot of research that's been done suggests it's through inflammation, but how? So I'm going to just talk about DNMT3A. It stands for DNA methyltransferase 3 alpha. It plays a crucial role in DNA methylation. So this is a process where methyl groups are added to DNA and they influence and regulate gene expression overall and cellular functions. So normal DNMT3A regulates which genes can be expressed. This is a highly simplified figure, but they actually place these methyl groups, or as my daughter calls them, me balloons, near genes or near places where transcriptional machinery can all congregate and allow for gene transcription or gene expression. But when these balloons are here, or methyl groups, CPG islands, the transcriptional machinery can't assemble. However, when there's a mutation, it's not able to methylate with high fidelity these regions, and so now there's dysregulated active transcription. And when we went back and looked at 81 patients in our cohort and looked at, so these are survival curves. So we looked at patients that had VEXAS that didn't have clonal hematopoiesis versus patients that did. We didn't see any real significant difference in overall survival. However, when we just limited, so when we separated them into just patients that had DNMT3A mutations and ones that didn't have, we definitely see a significant decrease in survival. So there was an association with DNMT3A mutant cases and a decreased survival. And we also showed that this didn't actually, so none of these cases were associated with an increased risk for MDS or AML. So inflammation is one of the most common manifestations in VEXAS. And it's been shown in multiple studies that UBA1 is definitely a driver of inflammation in this myeloid compartment here. But when we looked at, these are bone marrow cells from a VEXAS case, and these are normalized enrichment scores, so expression levels. These are subpopulations of cells. So we have the myeloid cells, and we have the innate B and T cells here. When we looked at the levels of different cytokines, we see that many different inflammatory pathways are elevated, but it's not just in the myeloid cells. These are the cells that have the UBA1 mutation, which is kind of at the level here. We know the T and B cells are not involved, but we see that there's an increase in inflammation in these cell subtypes also. So this suggests that maybe there's some non-cell autonomous component to the disease. So something else is influencing these cells to make the inflammation worse in these cases. The next two slides, I'm going to show some data from our lab, really exciting data. So this is Jenna Fernandez. She's a postdoc in our laboratory, and she did this data. We wanted to measure the influence of DNMT3A on these VEXAS cases. So we wanted to measure the levels of these CPG islands. So due to the role of DNMT3A as a methyltransferase, we looked at bulk DNA methylation. So we profiled these methylation changes in 19 VEXAS cases that didn't have clonal hematopoiesis versus 9 cases that did, and using an Illumina methylation array. So the plot shown here is called a volcano plot, and this is showing the differential change. The colored dots represent the statistically significant values, and each dot represents a methylation site. So this is a site right here of CPG islands. And the blue here, there's 4,000 different sites that are under methylated in the DNMT3A mutant VEXAS cases. So this is hypomethylated, which makes total sense because these, are DNMT3A methylated cases, mutant cases, I mean. But another interesting thing we found is if you use kind of a geographic roadmap algorithm and look at where these sites are that are under methylated, we see these are near genes that have pathways which are involved in differentiation of B and T cells. So adaptive immune response. Cytokines like IL-13 and IL-15. So we did an expression, a bulk RNA sequencing on this cohort next. So this is looking at gene expression. And here on the left is the same type of plot that I just showed you, but each dot now is representing a gene. So the red are upregulated genes in this cohort, and the blue are downregulated genes. And you can see of the genes here that are named, these are all involved in the pathways IL-13 or IL-15 that are indeed upregulated in our VEXAS DNMT3A mutant patients. So this validated kind of our observations on the previous slide. We also did site-seq. So this shows gene expression, but also has the ability to quantify cell surface proteins so that we can identify the type of cells that have the overexpression or the underexpression of genes. So it's a really cool type of technology. The plot on the right is also a volcano plot, but now the colors represent the different cell types and the dots represent the genes. So left is downregulated and the right is upregulated. And we know that the UBA1 mutations are restricted to the myeloid cells, but we see here in blue and in green, these are the B and T cells, and these are the cells that are upregulated. So these are the ones that are upregulated, so that have the IL-13 and they have the IL-15 pathways that are overactivated. So the adaptive immune cells are showing an increase in pathway activation, even though they're UBA1 wild type. So what's happening? So we think that the presence of this DNMT3A clonal hematopoiesis in VEXAS may be contributing to an even more exacerbated inflammation through an epigenetic influence on this adaptive immune response. So even though mutations in DNMT3A are seen in common age-related CH, if there's an inflammation present, the aberrant role of DNMT3A can aggravate and greatly influence the outcome and progression of a disease like VEXAS. And this is highly relevant finding that we can use to help us study MDS and MPN, also, that don't have VEXAS, but maybe in the context of our other types of inflammation or with therapies that, can prevent inflammation. And what I don't have time to show you today is the newest technology where we can now overlay, the map of the epigenome, individual methylation marks onto the genome to look at exact spots that are being affected by DNMT3A mutations and this increased adaptive immune response. So we can do targeted therapeutic interventions, to help with these cases. So in conclusion, VEXAS syndrome, kind of represents a new hematopoietic inflammatory syndrome defined by, with a propensity to become a myeloproliferative, myelodysplastic, or lymphoproliferative disease. This disease is driven by somatic mutations in the blood in UBA1, results in myeloid lineage predominance, and UBA1 in the context of clonal hematopoiesis, especially DNMT3A mutant cases, confers worse survival. But most importantly, the high prevalence of this syndrome, 1 in 4,000 in aged patients, and its only very recent discovery really underscores the need for more crosstalk and education across practices, for more multi-systemic symptoms. Recognition and ability to crosstalk and help facilitate the patient needs. A further investigation into the UPS system and the hematologic inflammatory linked disease, because I think this is very relevant and we're now seeing that. This helps underscore the importance of somatic contribution to disease, so acquired mutations. Why the blood is so important, it's because the circulatory system, can influence all other major organ systems because it actually flows through them. It houses, major parts of the immune system. It's exposed to all parts of targeted therapy for other organ systems. The blood is exposed as well as environmental exposures. So the blood is a very sensitive part and actually a very affected part and can contribute to other, very relevant disease and mortality in patients. And we also need more studies into causality and influence, of inflammation on outcomes in these patients and what can we do, as interventions with these cases. So I thank you very much for your time, and I'll answer any questions that you have. Thank you.
Awesome. Thank you. I love how, I don't know, when speaker is really excited, it definitely brings that. So thank you for that. I'm gonna go back, share my screen.
Let me unshare mine real fast. Oh, can I unshare?
Yeah, and we already have some people saying that it was really informative and it was, and thank you for breaking it down in an easy to understand way. So I agree on that. I agree on that. Thank you a lot. I also have here a question from a patient that it says, um, sorry, I understand VEXAS is caused by a somatic mutation, so thank you for that. And it's not inherited in the traditional sense, but should I still be concerned that now on I can pass this to my family, to my children?
No, you don't have to worry. So this is definitely not any genetic predisposition, or you don't have to worry about giving this to anyone else. No. Um,
Awesome. Okay, I have another one. Um, I have MDS and I've had some episodes that seem inflammatory. I haven't been tested for the UBA1 mutation. Would it be reasonable for me to ask my healthcare team to look into that?
yes, absolutely. I think that's a really great question because I know that there is a lot of outreach now in education, and this is a brand new topic. It's a very hot topic. And so I think there's a lot of institutions that are now recognizing this. We don't have VEXAS testing in a lot of places. And so, if I would definitely, there are some key clinical features of the disease that your hematologist or oncologist can look for right away. Otherwise, reach out to have your oncologist or hematologist reach out to Mary Lou and get in touch with me, and I can certainly help you with the criteria that they need to look for. But yes, screening, it's a non-invasive test, and it's something that I definitely would recommend because we definitely there's opportunities for you if you happen to screen positive right away. That there's opportunities now, trials that you can get involved in that are very effective. So recommend it.
Awesome. And now that we're speaking about that testing, can you explain to our audience, if you know, like a little bit of what kind of sample they need? What's the process like?
Yeah.
Do you know a little bit about it?
So our institution does a digital droplet PCR. It's just a basic blood test. So you would just in one of your routine blood draws that you go in for, just have them order the test at that same time, and then you don't have to get stuck again.
Oh, that's cool. That's for the drop, that's easier, I believe. So that's good to know. Also, I have another question. Managing two conditions feels overwhelming, and even more when they seem to be rare. So, what kind of doctors or team should I be working with to make sure I'm getting the right care? Is there a specific type of specialist I should be seeing for VEXAS, or can my hematologist or like oncologist handle it all?
I'm not sure I'll have the best answer, but for me personally, the team that I work with here at Mayo um, actually strive for outreach. So if there's any way that your local hematologist, even if you reach out to me, I can get you in touch with our team. But I think they're heavily involved in providing expertise and guidance with VEXAS. So Matthew Coster is our resident expert at Mayo Clinic, and he's who I would recommend to reach out for. I don't have experience outside of my, healthcare network, but
Yeah.
here at Mayo, I know we're very passionate about education and training and patient advocacy. So I recommend you reach out to Mayo because we have resources available that we can share with you, or you can be referred to Mayo Clinic too, which is also very highly recommended because we have seen a lot of these cases so far. And so I feel like we have, you know, we're right up there with one of the clinics that has the most experience with this. So I recommend it.
Thank you. I'll make sure to find that information on your website and share it with
I
our
can I can
audience.
send it to you.
Awesome, awesome. Thank you. I see this one and I think it's interesting. So you mentioned inflammation earlier. How is this inflammation from VEXAS different from other autoimmune or inflammatory diseases. So,
Ooh, another really great question.
yeah,
I
could be
really like this.
mistaken for something else.
Yeah, so VEXAS was discovered in 2020. And I can't concretely say that we really understand the differences or the similarities between inflammatory syndromes and VEXAS. So we're still learning the different signaling pathways and how this is, diseases like MDS are associated with high levels of inflammation as well. And so how is this related to VEXAS? That is another question that we're still trying to answer. So I think inflammation overall is kind of the same kind of phenomenon. So it's these myeloid cells, it's this aberrant dysregulation of microenvironment, and it definitely contributes to aggravated disease. So anything that you can do to lessen that will help, no matter which type of inflammation it is.
Thank you. And I see another one. Do we know how common it is for VEXAS to occur in someone with an existing bone marrow disorder? I don't know if you mentioned that already.
Yeah, so it's about 1 in 14,000. However, if you're older, a little older, then that frequency, the prevalence is a little more. So you can get to be in about 1 to 4,000 people. So it can be relatively common.
Yeah, and I'm so glad that we have like an easy testing to do.
Yes.
But you need to know what to look for when you're testing. Um, Also, let me see. I think I have another one. Okay, since VEXAS was only recently discovered, how can I be sure my healthcare team is even aware of it? So is there any ways to bring it up respectfully and make sure it's on their radar?
My advice is to be your own advocate. There is nobody, I mean, you've got to advocate for yourself. And so I would bring the question to your physician. You're not only helping yourself, but you might be helping another patient too by raising the awareness of your primary care person to this type of syndrome. Make them look it up. Make them tell you where you can get tested. Make them. I mean, they're there to help you, and you are your best advocate. I mean, nobody else. And so I give you the strength to ask them directly, very directly. I would be like, look, this is something that's very relevant right now. We don't know everything about this syndrome. I mean, it's could be related to other syndromes that we have yet to discover, and so yeah, it's very important to advocate for yourself, and good luck and do it.
Yeah, and even just mention it like, hey, I saw a webinar about this
Yes.
topic. Have you heard of it? Right? That might be a good one.
And I
And
can
I
send
see.
Mary Lou a
Mhm.
link to our Mayo Clinic resource page. I will do that.
Oh,
And so
definitely.
hopefully you can post it on your HealthTree link, and you can access it there. So that was a really good suggestion.
Yeah, definitely. We always wrap up an email for everyone with your slides and everything. So make sure we include that. And I see another question. So does a high um does a high an APO over 1800 have anything to do with VEXAS?
So not directly to my knowledge. Erythropoietin is so the erythropoietin receptor has to do with red blood cell, erythroid turnover. It could have to do with a lot of different things, but not directly to VEXAS to my knowledge.
Good to know. I think that's all the questions we have. I don't know, is there anything extra you want to let our patients know about VEXAS or anything else, just as a closure?
I mean, I just want everybody to know. So I'm passionate. I'm in the lab. I don't really get FaceTime very much with patients. And so when I get the opportunity, I really appreciate it. Behind the scenes, we're doing a lot. I think there's going to be targeted therapies, and I think they're going to be very soon available to patients. And so that makes me excited and proud and more passionate than ever to be able to, not only provide education about this so that more groups can get involved in testing and, all this information we're using, and this helps guide us and drive us to, which testing or what kinds of experiments we do, what kinds of therapies we focus on getting pushed through, so that they can be used in patients. So yeah, I mean, I just the ease that, I work with some of the most amazing people in the world that are, geniuses, and they're just to know that they're there and they're working on this, and that there is hope for everyone that suffers with these, really horrible. I mean, these cytokine inflammatory diseases. You just don't. And then these patients come in and give blood for research, and I mean, they should be commended because they're anemic, they're cytopenic, they have low blood counts, they're in pain, but yet they're there just so they can give their lifeblood so that somebody might get helped in the future. So I want to thank everybody for, you know, um, everything. And yeah, it was a pleasure to be here today. Thank you very much.
Thank you. I think there's someone who has another question. John, if you want to type it out or you want to unmute yourself and ask it away, we still have maybe a few minutes. Let's see if we can get that question.
Can you guys hear me?
Yeah, we can hear you. Go ahead.
Okay. I just have a question. So I'm 20 years old, and last June, am I allowed to ask a question about ALL?
I might not be able to answer it, but
Okay,
you can ask it.
Yeah,
okay.
you can ask it.
So in June I got diagnosed with ALL. I'm 20 years old, by the way, and this was last June, so it's coming up on a year. And then I went through induction therapy, and then 1 month of consolidation therapy, and then switched over to Blinn and became MRD negative. And I just started maintenance therapy about 2 weeks ago, I want to say, and I just started to get like this leg pain again. And when I got diagnosed, I had the leg pain. So it just always scares me for no particular reason. I got blood work done like a week and a half ago. And everything was like normal. But like I was just wondering like when I should be concerned if it is relapse. Because I also feel like I've been more tired than usual, but like it could all just be in my head, you know?
Yeah, I mean, I'm not an MD, so I can't give you medical advice like that. But I mean personally, I would check with, have you had molecular testing done for ALL specific alterations in the past? Is there something that they could look at for MRD recurrence?
I don't know if it's without a
It,
bone marrow biopsy, I'm not sure.
I would ask your physician if there's a blood test that's available that can.
Okay.
That's what I would do. And otherwise, for leg pain, that could be a lot of different things. It could be a deep vein thrombosis or, other things. I'm really sorry that that's happening to you, and I'm sorry I don't have a
Yeah.
clear answer for you.
No worries
Yeah.
at all. Thank you.
Yeah, John. And just so you know, if you're still there, we're having an event for ALL.
Ooh,
We're starting there. So in June 11th, we're going to have one. So if you want to drop your email in the chat, Marisol can help you register. And we'd
Perfect.
love to see you there. So thank you for jumping on. And I truly hope that that makes you feel like approach your healthcare team and ask that question directly. Yes, I will echo what Dr. Lasha said. Okay, well, again, thank you. That was really great presentation. Thank you a lot for allowing us your time and for learning a lot. So thank you again. Um, and yes, so for everyone who's still there in the call, I just want to let you know that, and a reminder that as you leave the webinar today, we would appreciate you taking a few minutes just to fill out a brief survey. That will help us a lot to just present more events that are really interested for you. And we would appreciate that a lot. We're having a few upcoming events that I wanted to let you know. We're having also in-person events. That's really exciting. And I'm gonna tell you about in the next slide. And also, I wanna let you know that we also have other topics that might interested. We also have financial events that are really interesting. The next one is gonna be on July 1st, and it's gonna be avoid Medicare Advantage traps and Medicaid eligibility pitfalls. That will be really interesting. And as I mentioned, in-person events, this year we were planning on visiting many communities. Connect to Curious is a free in-person event where you can connect with other patients, grab some lunch, learn from local doctors, and discover key resources from our HealthTree team. We already have official dates, and we already started a few days ago in Macon, Georgia, and we're gonna go ahead and into Peoria this week, and then Orlando, Harrisburg, Fresno, and so many other places. So if you live nearby any of these cities, please scan the code to save your spot. We would love to see you there and learn a little bit about who we are and what we can do for you. You can also contact our support team. So if you need any extra questions, they will be happy to help you. And before we wrap up, I just want to say a huge thank you to everyone for being part of this MDS community. Your support means so much for all of us, and we're grateful to have you here. I've included my email on this slide, so please do not hesitate to reach out to me if you want to say hello, if you want to say anything, any feedback. I appreciate that a lot. Once again, thank you. Thank you, Dr. Lasha, for being here with us. I hope everyone has an amazing rest of your day, and I hope we can see each other at another webinar soon. So goodbye, everyone.